Assalamualaikum warahmatullahi wabarakatuh. Thank you very much. First of all, let me introduce myself. My name is Lia Restimulia, and the topic of my presentation is the role of stem cell in allergic rhinitis. By the end of this talk, you'll be familiar with stem cell, particularly in allergic rhinitis. Allergic rhinitis is a nasal symptomatic disease due to allergen exposure, IgA-mediated inflammation. AR disrupts the quality of life, which is not only limited to nasal symptoms, but also activities and rest. So it is included as part of allergic rhinitis classifications. The prevalence of AR is 40% worldwide. In Indonesia, AR is in the rate of 24.3% and increases continuously. The incidence of severe AR is quite high, namely 67.5%. This is our study in 2018 that found moderate to severe persistent allergic rhinitis incidence about 60%. Dendritic cells as antigen-presenting cells after maturing with the help of interleukin 25, 33, and TSLP present antigens to Th0 with MSC class 2 and release interleukin 1, which activates Th0 to proliferate into Th1 and Th2. At the same time, APC produces interleukin 10, which can control the action of Th2 and regulatory T cells. Dendritic cells play an important role in allergic rhinitis, which has a high level of efficacy against Th. In addition, dendritic cells also produce interleukin 12 to induce interleukin production by Th1. Th0 cells differentiate into Th2 with interleukin 4 and TSLP, but this process is also controlled by T regulator with the help of interleukin 10 and TGF-beta produced by dendritic cells. Th2 cells induce B cells to become plasma cells by producing interleukin 4, 5, and 13 along with chemokines, thus producing IgE. Clinical symptoms that arise as a result of this process, including sneezing, runny nose, itchy nose, nasal congestion, and eye symptoms. The allergic process continues 6 until 12 hours after exposure. This process continues with infiltration and activation of effector cells such as eosinophils, Ts cells, and B cells within a few days, causing chronic allergic inflammations. The last 5 years research discusses the latest allergic theory, which is called the barrier hypothesis. This theory explains how allergens can damage the permeability of the tight junction in the epithelium, which can be caused by genetic that can produce reactive oxygen. The exposed epithelial cells to allergens produce the main cytokines in the innate response with arrhythmic stromal lymphopoietin, interleukin in lymphoid cells 2, interleukin 25 and 33. In the innate immune response, there are several other components that play a role: histamine, which can produce ROS. The increased inflammatory response to innate immune response is thought to cause an increase in heat shock protein 70 as a marker of oxidative stress. Immunotherapy is one of the treatment options which has the highest efficacy, but it's hard to make sure about the patient's adherence, and also it is still found that some patients do not respond to immunotherapy in the amount of 30%. This makes me wonder if there is any other potential to be used as therapy, and even better, that comes from the body itself. After reviewing many literatures, finally I decided to do research about stem cell or MSC. So we'll talk about the potency and stem cells. Potency along with faith, determination, and differentiation is one of those terms that you need to know and you need to understand in order to understand stem cells. To be a stem cell, you need to possess two main properties: the ability to self-renew, meaning you can divide, and divide, but at least one of the resulting cell remains undifferentiated. And you need to have the eye capacity to differentiate it to more specialized for when the times come. There are two main types based on the origin of stem cell: embryonic and somatic or adult stem cell, which is found in every person, for example, bone, skeletal muscle, blood vessel, and fats. Embryonic is called pluripotent stem cell only in embryo. Any stem cells that are derived from mature tissue in child or adult called adult or somatic stem cell, like multipotent, oligopotent, and unipotent. Umbilical cord contains stem cells, which are multipotent like adult stem cell. MSC is a multipotent stem cell, is in the form of spindle-like shape, which can be derived from bone marrow, adipose, umbilical cord, dermis, synovial membrane, and gingiva. The ability of mesenchymal stem cell to immunoregulate causes MSC to be widely used as therapy. This is the mechanism of MSC in asthma, whereas the secreted biological factors of MSCs play key roles in their therapeutic function. Many groups have begun to focus on the therapeutic activity. Du et al. Confirmed that MSC stimulated the proliferation and immunomodulatory ability of Tregs by enhancing the secretion of interleukin-10, TGF-beta in PBMCs from patients with asthma. All of these studies have confirmed the anti-allergic properties and therapeutic benefits of MSC in asthma. This is suggesting and promising future for their clinical use. I'm currently finishing my PhD, where my research was done on mice using stem cells from the umbilical cord. This is a nasal challenge photo and an observation of the clinical symptoms of the mice done by two observers. The stem cells used have passed the immunophenotype validation test and differentiation test and were declared eligible as treatment materials. MSC marker validation was performed using flow cytometry with SPM results able to express CD90, CD29, and negative expression of CD45 and CD31. Differentiation test shows that MSC can differentiate into osteocytes and adipocytes. MSC, this study is suitable as a material for treatment. This is our latest publications. I'm sorry for the limited time, so I'm not showing all the data, but in the results of the mast cell count, it appears that mast cell number is significantly lower in post-MSC treatment group, so with the plasma cell. But we found the increasing T regulator cell after MSC significantly. This is mechanism of MSC in many allergic diseases, whereas the immunomodulation function MSC exert an obviously suppressive effects on T cells, B cells, dendritic cells, and natural killer cells, and affecting innate and adaptive immunity, which play a role in allergic disease. Our study result can be explained by the ability of MSC to increase the T regulatory cell through interleukin-10, TGF-beta, resulting in the suppressing of many pro-inflammatory cells like T cell, plasma cell, mast cell, and others. Research in allergic rhinitis using stem cells is still a lot of work to do and a long way to get definite results. And in the future, it is expected that research can be continued to find a long-term safety, duration of response, the exact doses, and the technical feasibility of such cell-based therapies. Thank you for listening. If you have any question, I'll be pleased to answer them.